I mean, they are adding to the existing problem of classroom. So the students, they should be utilizing this opportunity. As I was saying to Arpan in the evening a little while ago, that while when we were students, we weren't fortunate enough to attend such conferences or symposiums, but you people are I mean should be utilizing this platform. Anyway, so the last session we have three speakers: Dr. Shamosri Bhattacharya, Dr. Rohan Gokhale, and Dr. Darren Tan. And we would begin by Dr. Shamosri Bhattacharya's talk. I would request Dr. Arpan Manna to give a brief introduction about the speaker, and then we can move on with the lecture. Arpan?
Yes, thank you, Dr. Prantik Banerjee, for beginning the session. So I am welcoming Dr. Shamosri Bhattacharya McCain, who is the currently Associate Director of Quality Control of Adigo Therapeutics in USA. As an AD, means Associate Director, Dr. Bhattacharya is responsible for ensuring manufacturing quality of Adigo's lead monoclonal antibody candidate for neutralizing coronavirus. Prior to joining Adigo, Dr. Bhattacharya worked as US Pharmacopoeia and PPD labs focused on the development and maintenance of the documentary and reference standards for a varied biologics portfolio that included vaccines, antibodies, insulins, and other complex products. She has been responsible for the development, validation, transfer, and life cycle management of analytical method for biopharmaceuticals for the preclinical through commercialization. Her scientific expertise includes structural biology and electrophoretic characterization of protein-based therapeutics. Dr. Bhattacharya holds a PhD in chemistry from the University of Wisconsin, Madison, a Master of Science in Genetics from the University of Calcutta, and Bachelor of Science with Chemistry Honors from Scottish Church College, Kolkata. So now the platform is over to Dr. Shamosri Bhattacharya. So Dr. Bhattacharya, please continue.
Thank you. Thank you for the introduction, Arpan. And I will be talking about my work over the last 5 years for manufacturing monoclonal antibodies and what is needed for it. So just give me a second while I share my screen. Can everyone see my screen?
No, it's not still visible.
It usually takes
Yeah,
a
yeah,
second.
it's visible.
Awesome.
Yes, it's visible.
Yes, sometimes the internet across the world takes a second. So as Arpan said, I am the Associate Director of Quality Control for a small company called Adagio Therapeutics that started last year in the middle of the pandemic. And for today's talk, I will be focused on giving you an introduction and a general background to myself, how I found myself in the field, what exactly is biotechnology and what I do in terms of the industry and global product development, and a general idea of careers in biotech. One caveat, my experience with biotechnology is very US focused. So I might not be able to answer all your questions if you have questions about the Indian industry. So my academic background, I grew up in Calcutta, born and brought up, went to Welland Goldsmith School for 15 years. And then I did my bachelor's study in Scottish Church College back of 2007. Arpan and I go back a long way. We were the same batch at Scottish Church College. After my bachelor's, I did the very Bengali thing of continuing on with my education for a long time. So I got my master's in genetics from University of Calcutta. And then I flew all the way to United States for my PhD. Went back to my roots in chemistry. My work was more in the realm of biochemistry, though, rather than sticking to straight up chemistry. in terms of my work background, my research experience has always been more biochemistry focused. In 2008, I had my first experience with research. I worked at CCMB Hyderabad for 3 months in glucagon production in common carbs, so Ruimach. In 2009, I was at Bose Institute for almost a year as a lab assistant. This was my gap year between end of my master's and my PhD. 2010 to 16, I was doing research for my PhD on crystal structures of bacterial phytochromes using X-ray crystallography.
Seminar.
In 2016 is when I entered the industry for the first time as a senior scientist. I was working at a small company called PPD Labs in analytical characterization of monoclonal antibodies. And then I moved in up the ranks in the same field. After that, I was at US Pharmacopoeia, and now I'm at Adagio Therapeutics. So what do I do in biotechnology? What is biotechnology? Biotechnology is application of processes at a molecular and cellular level to solve problems, conduct research, and create goods and services, so medicines, biofuels. I work with large biologics molecules, proteins that target a specific cause of illness. And it always starts with a living organism. That's the difference between biotechnology and a traditional pharmaceutical drug, which is a chemical substance that is manufactured synthetically in a lab. So I start with a mouse or a cell, something that is alive and breathing. In the United States, this is the traditional stages of what a biologics drug development process would look like. Things are in the discovery phase. This is when you're identifying your lead candidate, usually for 2 to 10 years. Then it goes into a preclinical testing. You test it on mice models or monkeys or bunnies in some case. After that, it goes into several stages of physical trials on human beings, usually 3 to 4, and each phase can last for a very long time. Subsequent to all of these phases, the FDA, which is responsible for approval of all drugs that come to America, will review and approve the product, hopefully approve the product. And then there's continuous post-marketing testing. So once a drug comes to the market, that doesn't mean the science stops. It still is the job of the company to ensure that every single batch that is being released is being correctly characterized. So typically, if you look at the span of this stage, it lasts for 10 to 15 years, costs a lot of money, upwards of $800 million, and each stage requires technical expertise from scientists. And all regulations are country specific. So FDA is responsible for the US market. I also work closely with European markets and Japan. Those are the biggest places we go to. So in the realm of biologics, my area of expertise is in manufacturing and quality control of antibodies. And what exactly are antibodies? We all have them. These are Y-shaped proteins that our immune system uses to identify and destroy foreign objects like bacteria and viruses. So when you get injected with the vaccines, as hopefully you will be soon if you haven't been already for your COVID vaccine, your body is going to produce an antibody specific to the coronavirus, which will help target the coronavirus, which we will call an antigen for this purpose, and bind to it, help your body destroy it by the antibody binding to this particular antigen. Now, using this mechanism that already exists, we are able to create it in lab, an artificial monoclonal antibody. This particular figure is taken from Seattle Times, and I'm happy to send this presentation afterwards if you want to look up what they were talking about. But antibodies in terms of treating coronaviruses are, instead of injecting you with a vaccine and having your body create the antibodies, we could specifically create a monoclonal antibody that already targets the coronaviruses and inject you with that, thus just moving across this entire process. And this is very important because right now, in terms of global vaccination, we are far behind what it should be. The manufacturing processes, vaccines versus antibodies, are very different. So we can still manufacture antibodies concurrently with vaccines. And the second thing to consider is a lot of people are unable to take vaccines for whatever reason. They're immune compromised. Their doctors have told them they aren't able to take it. In those cases, the monoclonal antibodies are very, very important. So that's what my company is trying to do, make a monoclonal antibody, which we've already made a monoclonal antibody, bring it to the market for it to specifically target coronaviruses. So in terms of the pharma, there's a variety of different functions that come together for a complete product to be put out to the market. So there's the basic research group, there's all your manufacturing groups, there's a whole section on quality right here, which you can see. So all of these are places where scientists fit in. Like every single part of this process is where science plays a big role in a pharmaceutical industry. Even support groups, legal, IT, finance, you need scientists to support a product and bring it to the market. It takes a village and a lot of years to bring a final finished biologics to the market. In pharma, my role is in global product development, which is a fairly broad branch. We are a team of scientists, associates, engineers. We develop the processes to manufacture the medicine, research what new techniques can be applied, optimize productivity, how to increase the amount of monoclonal antibody that we can get from the same starting volume, how to improve analytical testing or identification. And we work closely with other groups to solve problems. So what does a typical monoclonal antibody process look like? So at the very beginning, you have, I mentioned that for biologics, you have to start from a living source. So in case of a monoclonal antibody, it starts with one cell. So you have to pick the right cell for you to do your job. Next, you will take that cell and put it in what is known as a bioreactor. So the cell will multiply and create millions more cells. And each cell will be putting out the antibodies, the monoclonal antibody that you desire. The next step would be to harvest the monoclonal antibody from those cells. Then subsequently, you will purify it in order to throw away all the impurities that are also coming out of the cells. And then a lot of testing. This particular purification and testing is where my work falls under. So when we are thinking about manufacturing of a monoclonal antibody, we can divide the step into two parts. The very first step is selection of the cell, and that's the upstream part of the manufacturing. You have to determine what are you going to use to grow your cell. So this combines biochemistry, engineering, and physics to design a production process for this particular mAb that I'm working on. Usually we use mammalian cell lines, which means that they are not coming from prokaryotes. They are eukaryotes, non-yeast. Usually, the most common cell lines I've used so far is Chinese hamster ovary cell lines. These are then genetically modified in order to secrete the monoclonal antibody that you're working on. This is the first step in a chain of what is known as good manufacturing practices, where you have to follow very specific regulation that has come out of the FDA, the EMA, the Japanese Pharmacopoeia, to ensure that there is no contamination of your cell line. Because if your basic starting point is contaminated, then what you're going to get in the end is never going to be a good product. The upstream development works really closely with all other teams in order to ensure they are selecting the correct cells and the correct process for this particular, whatever monoclonal antibody you're working on. So what is happening here? You start with a single cell that's producing maybe 2 monoclonal antibodies, and then you let it thaw out, you give it the correct conditions, and the cells multiply. So now you have thousands of these cells producing thousands of molecules of these monoclonal antibodies. So the goal of upstream is to grow enough cells to express your target protein. These cells are grown in what we call bioreactors. This is a massive setup. So it starts with this tiny vial, this tiny vial right here. And then we take it into a round bottom flask, then a bigger flask, then multiple flasks. And then we put it into the setup that can take over a whole building to produce enough monoclonal antibodies for a cell expansion. And you continuously spin these cells, give it the right conditions. And by right conditions, I mean, what do cells need to grow? They need nutrients, usually glucose, oxygen, water, proper environment, right pH conditions, right temperature, enough oxygen, and agitation. It's similar to making bread. I know that's not a very good comparison because I never made bread while I was living at home, but think of it in terms of the chemistry of making bread. You need yeast, you need your basic nutrients, your flour, sugar, water, and a proper environment. And the scientists have to choose what they mean by proper environment. The pH conditions, the dissolved oxygen, all of this is input that we get from the lab scientists who work there. So now we have the ideal conditions for our cells to grow. Next, we are going to track how much, how they affect our final amount of monoclonal antibody production. So here you see a comparison of pH. So at different pH conditions, depending on which day I'm looking at the data, the highest amount of production is pH 7.1 and day 10. So you're charting everything out, being very systematic about the whole process to identify exactly the right conditions, which will give you a pure and the most amount of product that you need to continue on with the process. So the next step, now we have all the map that we could ever need to continue on with the process, is to figure out how we can purify our monoclonal antibody. So the downstream process development team is responsible for designing and transferring and supporting all purification processes. It could be for clinical samples that are going directly to the clinics for the phase 1, 2, and 3 trials. It could be for commercial samples. This might be already an approved product, and so you're manufacturing enough to supply to the US market, the Indian market, what you have it. The downstream team bridges the gap between research and manufacturing because they need to understand enough about the research to develop a scalable and cost-effective purification processes. Because you know what happens when a company can't make money on the drug? They will stop producing it. So it's very important to keep cost in mind as you are manufacturing any sort of pharmaceutical product. So the basic function of any purification process: remove what we don't want, which would be in this case any other protein, DNA that is produced by the cells. If there's aggregates, the molecule might not have correctly formed its three-dimensional structure, so it might be stuck to itself, as well as all other ingredients that came with the cell culture media, or your glucose, your whatever nutrients, your yeast, whatever you fed it. Next, you have to control growth of any bacteria. Cell culture always comes with a growth with that caveat. Are there bacteria that are growing that will impact your final product? Because if there is bacterial contamination, we are not able to release the product to the market. So this is known as a bioburden control, and there's testing, extensive testing around making sure there's no contamination by any other living thing in your product. And the key to this entire process is reproducibility, because it's possible that I'm manufacturing this in a plant in USA, and then day after tomorrow, we get a plant in Japan, and they will have to repeat everything that we did here and get the exact same result. So the principle of separation is ion exchange chromatography. In theory, it's really simple. It's based on negative and positive charges are attracted to each other, like repels like. So you have your monoclonal antibodies, which are usually have a slight positive charge, and then you will throw them through in a flow through mode through an ion exchange chromatographic column, and which if it's a positively charged column, you will expect it to stick to it until you wash it down with a buffer that will release it from the column, or it could be a negatively charged column, in which case you would expect the entire flow through that's coming out of it to be the impurities. So impurities in this case, as we mentioned, can be your cellular debris. It could also be living things like DNA and bacteria. So this is a very important step, and figuring out what kind of column you need is also very critical, and it depends on the product you're trying to manufacture. So the downstream process is also requires a large facility. So both upstream and downstream processes are usually housed in the same facility, and it needs significant amount of filtration. You're filtering your product and to ensure maybe bacterial cells, which are a different size from just an antibody, you are able to separate them, and then several chromatographic steps to remove all the impurities in a step-by-step manner. Next, after now we have our manufacturing part completed, the process doesn't end. Now it's the analytical sciences to step in to confirm that what was manufactured is what we want to be manufactured, and also confirm the purity of the product. So just for scale, in terms of what we are looking at, a hydrogen atom is 1 atomic mass unit. Tryptophan is 202. It's 1 amino acid. A large molecule would be insulin, which is about 5.7 kilodaltons, or 5,734 atomic mass unit. We are looking at manufacture of an antibody, which is 150,000 atomic mass unit at 150,000 kilodalton. So size guides analytical process development. How you will separate a tryptophan is very different from how you will separate a monoclonal antibody. Even within the antibodies, there might be different sizes that you're looking at. So analytical process development for identity confirmation and purity determination is heavily guided by the size of the product we are looking at. So product characterization, what does that entail? You perform a physical chemical characterization, and you have to provide this data to the FDA, and they will review everything, make sure that the product is identified to what it is. They will validate the manufacturing site where it was produced, because if something's wrong with the manufacturing process, then the final product is contaminated and you cannot inject it into people. The characterization group also assists with qualifying what you are comparing things to. So that's your reference standard. And that's a whole different process of identifying the correct standard to compare it to. And I could give a whole separate presentation on it. But just keep in mind that there's a lot of functions within science that are involved in it. So there's process development, there's research and development. There's bioanalytical, which means that you will be using LC mass spectrometry to confirm sizes. And these are very critical steps in this process for FDA to approve it, because if your drug isn't approved, it doesn't matter how much work you put into it. So the analytical sciences really supports all development groups from the very beginning through the upstream, through the downstream. You have to keep in mind what tests you can do to confirm the identity in order to even start producing the monoclonal antibody. So there's assay development, there's you get these samples, you analyze them for cell culture purification groups, there's clinical candidate, there's a lot of work to be done. And this analytical sciences in United States employs thousands and thousands of people. Usual tools of the trade for us, a lot of pipetting and a lot of chromatography. So we do a lot of cell culturing. We run a lot of size exclusion chromatography to correctly identify our monoclonal antibodies. Now that we have confirmed the identity, we have made sure that our product is pure. The next step is to figure out how we are going to get it into patients. This really is the differentiator between different products. And that can be a huge technological advantage as you enter the market. So would you rather take a vaccine that's a shot versus something that you can is a nasal spray? That really affects and this differentiates your product as you come into the market. So the drug delivery and device development is a very, very crucial part of the development process. And again, this isn't exactly a stepwise thing, even though I'm presenting it that way. All of these groups work in tandem because how the drug is going to be delivered might affect your manufacturing process because to give you a very quick example, for an injection, for a monoclonal antibody to be injected into you, the concentration needs to be 150 mg per mil or higher. For it to be an IV infusion, to put it into an IV bag, which is then injected into a patient, that only requires 100 milligrams per mil of concentration. So when we go into the manufacturing process, if you don't know that, then you would have to go back and remanufacture the whole thing because you can dilute a product. You cannot really concentrate it from something that's 100 mg per mil to 150 mg per mil. So just some things to keep in mind as you come into the device development and figure out how you're going to administer the final product. So that was the overall idea of drug development. Next, we come to what does it take to have a career in the pharma industry. It doesn't take a whole lot of degrees. It depends upon which level you're going to step into. I work with people with high school diplomas, with bachelors, with masters, with PhDs, doctors, with PhD and 10 years of postdoc experience, spans the whole spectrum. There's space for everyone. Since this target audience is mostly bachelors and masters, I will focus on the BSMS and PhD MD parts. I work with people with a variety of degrees: chemical engineering, chemistry, biochemistry, molecular biology, cell biology, statistics. My husband is a biological engineer. We work in the same field. And a lot of the times you can get your foot into the industry through a summer internship. These sometimes are paid. They can also be unpaid. But that really is a good way to build a relationship with the company you want to work in and start your career there. Positions usually start at the research associate level here or a research scientist. They can pay anywhere between 40 to 110,000 depending on what degree you're coming in with. But there are additional benefits. There's bonuses. They contribute to your retirement. There's stocks. They also sometimes pay for you to go back to school, go back and get another degree. I had a friend who worked for a company and they gave her 3 years of leave and paid some of her expenses as she got her PhD in Europe. My husband is currently getting his MBA, which is paid for by his company. I stepped in after my PhD without postdoctoral experience. I have not seen a postdoctoral experience add a whole lot to this. It's not necessary if you want to come into industry anyway. Starting salary somewhere around 60,000, and then you rise to the mid-management level and senior leadership. There is a wide range of salaries here. It really depends on where you're living in America. Cost of living differences are major. If you live in New York, they'll pay you a lot more than you live in the middle of nowhere, Montana. And the same additional benefits, I could still get another degree if I wanted, which I'm not going back to school ever again. They contribute to retirements, you get stock options and bonuses every year, depending on your performance. So having said all of that, this is not all the options in terms of careers for people with a degree in chemistry. You can go into drug discovery, but there's also teachers, robotics, materials chemists are highly valued as robotics engineers, astronauts. I have several friends who got their PhD and are currently environmental scientists. You can go get a degree in pharmacy and work at a work in that section of manufacturing. Patent agents are often scientists. A lot of chemists I know are either patent agents or patent attorneys. Management consulting, I have a friend who works for McKinsey because they need people with science expertise to go help their clients. Power plant engineers, aerospace, medical writing is big. I have a lot of friends who are who are editors for Nature or Cell or Science. So definitely use LinkedIn to research and network if you're particularly interested in a company. If you want to talk to someone specific, write them a note on LinkedIn. Don't just send them a friend request. That's creepy. And having said all of that, I'm going to pause here and take any questions.
Yes, students, any questions? That's some insights from someone who has been there, done that. So
yes, I am happy to answer questions about how I came to America, what the PhD program looks like, or how I got into industry.
Yes, students, please. I mean, you can ask questions not only from your subject, but how she ventured out from chemistry to genetics, back to biochemistry, and now where she is now. This has been a journey. So I mean, you should be somewhat inquisitive about different stages of this particular journey.
And if you can't think of questions, that's fine. Feel free to email me later. That's okay.
Yeah. Yes, and
there are
as
some
she
question
said
in the chat box. That ma'am, you have mentioned about 10 to 15 years of processing regarding the vaccine to be launched in the market. But we have seen that Pfizer marketed COVID vaccine and Russia market Sputnik vaccine within 9 months for the research and launching it in the market. Can we rest assured about the quality of the vaccine?
Yes. So the reason it takes that long is because the review process is that long. Clinical trials take months and years in order to even set up. So what happened is because of the pandemic, all the money got focused into development of vaccines, and things that would have taken a very long time just because of money or red tape or bureaucratic reasons went away. Poof, they were gone. So now the entire process was a lot simplified. All the manufacturing capabilities have been devoted to COVID-19. Finding a manufacturing plant pre-COVID for a vaccine, very difficult. There were very few. They were not qualified by the FDA. So imagine all of a sudden the entire world focuses on one problem. This is what we can do, bring a vaccine to market in 9 months. So think about climate change. If we all focus on climate change, we can solve it. That's the power of the human brain.
Very good answer. So another question by Dr. Devapratim Das. He's our faculty also, and also very good organic chemist.
Okay.
So why USA pharmaceutical market is 1,000 times more promising than Indian market?
I don't know if I have a good answer for it, but there are a few things to consider. First of all, science funding in the United States is very, very high. There's a lot of money that goes into making sure that the pharmaceutical market is sustainable, and people are able to manufacture here, and people are able to develop cutting edge science here. So I think we are talking about a difference of scale. It's a huge difference of scale right there. And that's not saying anything against India. Keep in mind, US has been a country for over 300 years. India has only been a country for a little over 70 years. So eventually we will get there, but it's very hard because it's a small country with a huge population pressure. Where is the space for a big manufacturing facility? So all of that has to be kept in mind. There are significant differences and pluses and minuses. Besides a generic answer like that, I have no specific background in Indian pharmaceutical industry. So I'm not able to give you a very well-rounded answer.
Thank you, Samastri.
There's
Yes.
another interesting question, that is, we are acquainted with the term placebo. Does it happen with monoclonal antibodies too?
I mean,
Well,
placebo as an effect happens with everything, right? So, but in order for a monoclonal antibody to be approved or any drug to be approved and come to the US market specifically, there has to be a clinical difference. It has to be way higher than placebo because there is already a control group. Every single clinical trial has a control group. So the FDA has very strict regulations on how high the control group versus the group that actually gets the medicine, what the differences are going to be before it comes to the market. So of course there's a placebo, but it is not clinically significant enough.
Sam, may I ask a question, sir?
Yes, yes.
Sure.
Yes, sure.
Thank you very much, ma'am, for your wonderful talk. Actually, ma'am, my question is regarding the monoclonal antibody. We know that in this current scenario, the COVID-19, that is SARS-CoV-2, attaches its spike protein with the S2 of the host cell and enters into the cell through receptor-mediated endocytosis. So here, vaccine plays an important role to boost the immune system. So I have seen, though the number is very less, few people after being administered with the Covishield and also after being with the virus, they have got the again they have got infected with the virus. So is it that the new virus or the mutant one is embedding through our immune system, or what is the process that after being administered with the vaccine or something, they are again being attacked with the virus or being positive?
So there's a lot of nuance to that answer. So I'll try to break it down. There are two ways this can happen. The really bad way this can happen is viruses constantly mutate. That's an everlasting constant thing about viruses, is they will continue to mutate, continue to change, continue to escape your body's immune system. And when you have a situation where, say, 10% of the population is vaccinated, you are creating an artificial selection pressure. So the virus is going to try to evade that. And that's the worst case scenario. Now you have a virus strain that will escape the vaccine completely. But that's not what is happening here. What is happening here is a lot less dangerous. People who already have the vaccine, depending on the efficacy of the vaccine, they will evade the very severe consequences of COVID. So the virus might still come in, they might still attack your immune system, but you have a lot of protection from the vaccines. Yeah. So you might get coronavirus, but it will be like a mild case of influenza. Yeah.
Thank you, ma'am. Thank you.
Yeah.
Samastri, there is another question. At present time, from Sneharko Poria, at present time of the global vaccination drive, all the vaccine manufacturers are going all out to attain the higher efficacy. As a layman, should we really be concerned about this efficacy, and how is this efficacy is calculated for a particular vaccine?
There is a calculation for the efficacy. It is done during the phase 3 clinical trials. You look at your placebo group, which didn't get the vaccine. You look at your group, which did get the vaccine. And then you calculate the difference of the number of cases of coronaviruses that have existed in these two populations. So it's a comparative study. That's the easy mathematical answer on how to calculate efficacy. Now, the question is, everyone is going for the highest efficacy. Of course, efficacy is very important. But what is also important is it broadly neutralizing the coronaviruses? You could have a very efficacious vaccine, and it only neutralizes like one strain of the coronavirus, doesn't work against any of the others. Even if it's 100% efficacy, it doesn't really make any difference in that case. In America, the FDA had set a very basic ground rule, and that's the ground rule for most vaccines that exist, that anything 60% and above is considered good enough. But there are two angles to consider when you look at efficacy: the number, and is it working against a wide number of strains, because all over the world we are in hundreds now in terms of strains of coronaviruses, so it has to work against all of them.
Okay. Another question is that the second part of the question is that for monococcal antibodies, in the same way, efficacy is calculated like vaccine.
Yes, efficacies are usually calculated in the same ways, but vaccine efficacy is an actual number. It's always available to the public, not just for coronaviruses, but for anthrax or polio or all of the vaccines. For monoclonal antibodies or even drugs like aspirin, the efficacy is it clinically significant? If it's clinically significant, it is released. And there's subsequent research done to see if it's clinically significant against like all people, or does it only work with immunocompromised individuals, or does it only work with a certain subsection in terms of ethnicity? So it's a very different calculation compared to vaccines, because vaccines usually are approved for worldwide use, but it has the same mathematical basis. Yes.
Yeah. Another question is very pertinent question in this situation by Ipsito Prasad Pal. Ma'am, considering the current situation, we are have not practiced any practical chemistry for about a year now, both in BSc and MSc. Is it some way or the other affect us if we opt for industrial career?
So as someone who hasn't stepped into a lab in 2 years, I am sure it will be hard if suddenly someone actually gave me an experiment to run. I run the people who run the lab now. There is no really good answer to this question. Probably is as you step back into lab, you will have the muscle memory doesn't exist again. You will have to start at the basics and you will have to learn it. But keep in mind, your brain is active. You're taking in all this information, and that's what companies hire you for: your ability to think critically through a problem, your ability to arrive at a solution. So don't undervalue what you already have. You can relearn the muscle memory of how to use a pipette, how to correctly weigh things, but the critical thinking skills is what is going to sustain you through a very long career. Arpan, শুনতে পাচ্ছি না।
Yes, sir. Yes, sir.
Sir, please.
Doctor, yes, sir. Please,
Yeah.
please.
Yes, sir.
Professor Ghosh will ask a question. Yes, sir. I think there's some connectivity problem on his side. Okay.
There is another another question that is coming from Roji Banerjee. That how was the transition from chemistry to genetics? Was it frightening or challenging in a good way?
It was challenging in a good way. The first semester was very hard at University of Calcutta. I didn't do as well as some of my classmates who came in from microbiology or biochemistry, but by the end of my 4th semester, I was doing as well or better than them. There is a lot of benefit from coming into genetics from a chemistry point of view, because you understand the molecular interactions. You know what it takes for a DNA to come together at the very atomic level. So I didn't have that. Hindsight is always 2020. I didn't have that viewpoint at the time. I was just looking at the numbers on my results. I was like, this is not good. So I had to study a lot, but it ended up fine. And really, the results only matter up to a certain level. It's about how much knowledge you retained from your programs.
Okay. Sir, are you there? Professor Ghosh?
Yeah, my connection was on this time.
Yes, you are audible,
The
sir.
connection. Yeah, you're audible now?
Yes,
Okay.
yes, yes.
Thank you, Samasri, for the nice lecture. I want to know, we are hearing about nasal vaccine. What is the progress in that field?
I don't think it's in clinical trials yet. They are still developing it. They did try the nasal vaccines with the flu vaccine, but the efficacy dropped So that's one of the big reasons that they were unable to keep it going for the flu vaccine. So we'll see what happens with the coronaviruses, but it's still in a research and development phase. But everything is moving so fast. If it works, you will probably see it in the market in less than 6 months.
Samosri, Professor
Okay.
Ghosh.
And next question. Can you hear me?
Yes.
Yes,
Bakun,
sir.
can you hear me?
Yes, we can hear you, sir.
Okay. Next question is. What is the? Wait. Yes. Let's see.
Hello, sir. Hello. Samosri, Professor Ghosh is the professor
I
of
think
our
he went to. Yeah.
professor of our Anasen.
Acha.
Anasen was the student of Professor Ghosh.
Oh, I feel like I've heard his name
Yes.
before.
Because in presidency, actually, we retired
Yes.
as president.
Okay. I see one more question as we are
Sir.
waiting.
I
Huh,
hope sir had
yes,
come.
yes, and-
Sir. If you have seen any other question,
No.
Samosri, just tell.
Hello.
Sir.
Yes.
Sir, please tell.
কোভিডের
Hmm.
সময়
Please tell, sir. You are audible.
Yeah. I want to know from Samsri. Can you hear me?
Yes.
Yes.
Okay. After 2 doses of vaccine, how long it will take to produce antibody in our system?
2 weeks is what the FDA has said.
So can people test for the antibody and get some information about how much antibody is produced and things like that?
Yes, you can definitely get tested for the antibodies. I am not sure what the clinical level would be. That's peer-reviewed, publicly available information. So the Lancet paper will probably have a good number for you there. But you can definitely get tested after 2 weeks. And if you have antibodies, what level they are at, they will tell you. After a certain it depends on the test you're doing, but a lot of these tests don't go for precision. They go for if your antibodies are at a certain level, they will just give you a check mark or a plus sign. That's usually how I see my antibody levels here displayed in America. So keep that in mind, but definitely an antibody test would be able to reveal whether you have anti-COVID antibodies or not.
Okay. Thank you very much.
No problem. Thank you, sir.
Thank you.
Okay. So Arpan, I don't think there are any. There are other questions.
Okay. Thank you, Samosri, for wonderful talk, and also wish you all the best for your wonderful journey.
Just
really
just a note that regarding that question, how was the transition from chemistry to genetics? Dr. Bhattacharya may have been somewhat fortunate, because those days almost all the faculty members in the Ballygunge Science College in biochemistry, genetics, or biotechnology, they had their background of chemistry. So
actually,
that may have been somewhat
genetics
helpful.
department is housed within the Department of Botany. So I learned a lot about plant genetics in my 2 years. But you're right, biochemistry
Now,
under University of Calcutta.
Yeah, but genetics now is an independent department.
It is.
Now genetics is an independent department.
Yes, it was a great program.
Anyway.
And I did get into both biochemistry and genetics. My deciding factor was the class size.
In this regard, Samosri, I want to mention that Calcutta University, UGC has done a great job that they have almost eliminated chemistry from the biochemistry syllabus. The really most fun part. Myself and you probably know Dhrubajyoti Chatterjee.
Yes.
When we started the biochemistry course, we have 50% chemistry, 50% biochemistry, some spots, and they said that past chemistry at the past level will be enough to go for higher biochemistry, which I don't agree. So
That's
this is
an
the funny
interesting
part in India
viewpoint.
now.
I don't agree with it, but everyone has their views.
Acha.
Yeah.
Samosri, one last question by Anamika Saha, that how long the vaccine will work for COVID-19?
We do not know the answer to that question yet. Keep in mind that the vaccine has only been in market for 6 months. So at 6 months, which is now, they tested and found that the antibody levels are consistent. The companies are already working on booster doses at a 12-month interval. So until we have a concrete answer, we will have to get booster doses every year, same as your flu vaccine.
Probably
Just on
I
the flu shots, this will be.
Yes,
Yeah.
if you have something.
just on the regard of the last question, I would probably I have a kind of confusion with the advent of all these new variants of concern that probably we are all again concerned with. So, Dr. Banerjee, how do you feel that is the present antibody or the present vaccinations are good enough to restrict? I mean, prevent those. The critical symptom developments.
That's what research has shown so far. They tested the Pfizer and the Moderna vaccine against the South African variant, the double mutant variant from India. It is still able to restrict all severe cases of COVID. So even with all the new variants, there is no reason not to take the vaccine.
Yeah, one last question, Samosri, just. Yeah, how do you think about flu shot or pneumonia shot after the 2 doses of vaccine? Will it be effective? Or what gaps I should give, for these 2 shots after 2 doses of vaccine shot?
I
Will
believe
that be
the
effective?
official
Pneumonia shot and
guidance.
flu shot?
Yes, I believe the official guidance here in America is at least to wait 2 weeks because your body is still, your immune system is still on overdrive from the COVID vaccines. But after that, you can definitely take the rest of your vaccination. So really, you can go away about your normal business and get your vaccines, do your work, go out. Right now, they've said in America, the CDC has said that if you have both your shots and you've waited 2 weeks, you can be outside and not wear masks. So hopefully we will open up soon and India will get vaccinated soon. We need it.
Okay, thank you.
Thank you, Samosri. It was really very great talk actually, and you had talked about vaccine, which is a burning part, burning story actually nowadays. Thank you again. So be with us. So I am transferring my mic to Dr. Prantik Banerjee, who will introduce our next speaker. Prantik.
Yes, thank you, Dr. Aman. Yes, this vaccine issue is burning both metaphorically as well as literally nowadays, at least in our country. Anyway, so after such an insightful session, we will move on to our next speaker, Dr. Rohan Gokhale, and he is a research
Prantik
scientist at Nissan.
Prantik
Yes.
Darrenton. Prantik
Rohan
Darren.
Gokhale,
Darren. No,
Okay,
not. Darren will.
Okay. Okay. So I'd like to
Rohan
apologize for
is
the
unable to
inadvertent error.
present today actually due to some problem.
Okay. So in his absence, we would move on to our next speaker, who is Dr. Darren Tan, who is the Chief Development Officer at the Unigrid San Diego, California, USA. He's a co-founder of Unigrid LLC, which is an energy storage company, and which deals with the cutting-edge technologies in battery, solid-state batteries. He was previously a lead researcher at solid-state battery development work at University of California, San Diego, at the Sustainable Power and Energy Center. So I'd like to invite Dr. Tan to, yes, thank you, Dr. Tan. Over to you.
Thank you, Pat, for the wonderful introduction. Good evening, everyone, and also good morning to those who are in America. And thank you, Samosri, for the wonderful talk. And I also learned quite a thing or two about vaccines. So we're going to switch the topic a little bit towards energy and sustainability. So let me share my screen quickly before I get started. Okay. Okay, can someone confirm that you can see my presenter mode?
Yes, yes, yes. It is. It is.
Yeah,
clear.
yes, sir, you are.
Okay, great. Okay, just to briefly introduce myself, my name is Darren and I'm a co-founder and also as introduced, the Chief Development Officer at Unigrid Battery, and we are seeking to become the next generation of distributed energy storage solutions. So we are not talking about mobile phone devices or laptops, we're not talking about electric vehicles. Here we are concerned with the grid, the electrical grid. As a brief introduction, Unigrid's aim is to capitalize on the very massive untapped grid storage market today. We know that demand for renewable grid energy is silently but rapidly growing, and this is universal throughout the entire world. And wherever you have renewable energy generation, there must also be storage. Unfortunately, all the lithium-ion batteries in the world, or all the lithium that exists on planet Earth, even if made into batteries, will never be enough to meet this demand. Today we hear a lot about lithium-ion batteries powering electric vehicles, but the reality is that all the global lithium supply amounts to only about 15 million tons. And that may comfortably serve the electric vehicle market for the next few decades or so. But we need a vastly more abundant alternative, something like sodium, which totals more than 3 orders of magnitude higher than the lithium supply in order to capture this giant grid storage market. On top of that, grid storage batteries also need several other requirements, such as high volumetric energy density. They need to be absolutely safe. They cannot use any expensive elements at all. They need to exhibit extremely long calendar and cycle life, and also be able to operate over a wide operating temperature range in order to cater to the needs of people in the cold and also warm climates. All these combined requirements are things that existing lithium-ion batteries fail to meet. So global supply of lithium isn't the only problem. Lithium-ion batteries are currently too expensive for the grid. This is mainly due to the high cost of raw materials. In lithium-ion batteries today, they are often based on both lithium as well as unwanted rare earth elements such as cobalt, nickel, and sometimes other kind of element dopants as well. Compared to more abundant elements such as sodium or iron or things that you commonly see and use, lithium and cobalt are extremely expensive and they are orders of magnitude higher in cost. Additionally, when we consider batteries for the grid, the battery prices start to play an even larger role, because as cost fraction of the grid is much higher than smaller devices or even electric vehicles. To give an example, when we buy an iPhone, it may cost about $1,000 in USD, but the cost of the battery is less than $50. So it's actually only less than 5% of the entire device. Likewise, in an electric vehicle, the cost of the battery pack is maybe 20, sometimes at most 30% of the entire electric car. But when we look at grid energy storage, where we use stationary batteries, the battery cost makes up the largest component of the device, because it is the main device itself. It's meant to serve, it's not meant to transport people, it's not meant to send messages and things like that. So we do want batteries to be as low in cost as possible. The second concern is also that of battery safety, and this is something becoming something of a major issue. While we occasionally hear of some phones or laptops catching fire, perhaps 1 or 2 electric vehicles burning on the street, and we may have seen YouTube videos on things like that before, they are still considered relatively small fires, which are somewhat manageable with reasonably low risk. Our existing firefighting infrastructure can deal with this type of fires. However, the consequences of a potential grid battery fire, which is many, many times larger than battery packs in small devices, can be quite catastrophic. A grid battery cannot tolerate the same degree of safety hazard, no matter how slim it is. To put things in perspective, 1 in 10 million phones that are produced in the world are expected to catch fire. So there is a 0.001, a very low percentage chance. But when we deal with that in the grid, then it becomes a lot more dangerous, and we cannot tolerate the same degree of probability. Finally, we also have a concern of battery waste. If we are going to adopt batteries on a scale much larger than what we have today, how are we going to ensure that it is sustainable? And if we don't address battery sustainability before it even hits the market, we might undo a lot of the environmental benefits we seek to achieve in the first place. So what is our solution in Unigrid? To this end, we are proposing the use of a sodium or solid state battery to address these problems. Now, compared to a lithium-ion battery, the chemistry itself is quite different. Firstly, in lithium-ion batteries, it is often comprised of a transition metal-based cathode, something that contains lithium, cobalt, nickel, often manganese as well, and oxygen. And you also have a liquid electrolyte to transport the ions, as well as a graphite-based anodes as a counter electrode. Compared to this traditional chemistry, we instead completely eliminate the lithium, cobalt, nickel, and other expensive elements from the cathode in order to keep costs low. We also replace the flammable organic liquid electrolyte with a highly stable solid-state ceramic. So in other words, it is non-flammable at all. Finally, we also replace the carbon-based anode with a very dense, or sometimes we call it dendrite-free sodium alloy, which allows it to take up much less space. So overall, this gives us the ideal combination of high volumetric energy density, absolute non-flammability, so we dramatically reduce risks, and it is much lower in costs. It also improves its longevity because it's a lot more stable, and it's also able to operate over a wider temperature range, so something like about -20 to 80 degrees or so, which is what we have tested. To add to this, there are also several unobvious benefits when we move towards solid-state batteries in larger devices. Because solid-state batteries are intrinsically safe and can operate over the wide temperature range as described, we can reduce the requirements of thermal management as well as safety reinforcements. Because in a typical device, the battery or the motor or whatever device we're currently using often needs to be very thoroughly managed by some kind of thermal management device. We have to keep batteries cool enough to ensure that they don't overheat and result in thermal runaway. But when you have a battery that is entirely solid and ceramic, you really don't care what the temperature is. And you can allow it to self-heat and operate under any condition we want. Sometimes we often forget that the energy that is used to cool the battery pack in an electric car comes from the battery itself. So that is definitely have some consequence on the energy efficiency and the driving range or the utility of the entire device. Secondly, when we move from the materials to cell, the pack, the system level, there are also additional benefits when we use a all solid state battery. Firstly, things are a lot more densely packed. We don't have to have very elaborate safety reinforcements or elaborate cooling systems and things like that. So overall, we can pack more energy within the same volume of space. So this is one of the first devices we made so far. This is a picture of me in 2019. I was not wearing a mask at the time because this was pre-pandemic. But this is just a video of when we cut our all solid state battery under operation. So currently it's a small single layer device, something like half the size of a credit card or so. And when we cut it, it still remains in operation. There's no leak or anything of the liquid inside because there is no liquid, and it's extremely safe. It doesn't catch on fire and things like that. Now, I highly recommend you don't try this for regular lithium-ion batteries. This is another video of us putting it on a hot plate at about 200 degrees Celsius. If this was a lithium-ion battery, it would swell very dramatically, and we would see unpleasant things start to occur. But in this case, it just sits there and do its job, right? To put it simply. Yes. So in other words, we show that it's very safe, and we can show that it operates over a wide temperature range. Now, recently, there has been a lot of news in the media about solid-state battery companies and how are we any different. So here's a table summary of some of the major solid-state battery companies that operate off the United States. And we can see that almost all these major competitors focus on lithium-based chemistries, which is not surprising because their target markets are electric vehicles. In Unigrid, we instead focus on sodium, where its combination of low cost, good safety, and longevity is ideal for grid storage applications. Secondly, we also adopt all solid systems with absolutely no liquid in them. Most companies still use some kind of organic hybrid. And we can achieve this because we adopt a quite novel halide-based solid electrolyte, which no other company has used before. This unique solid electrolyte combines the stability of conventional ceramics and conventional glasses and processability of glasses to improve the performance and also demonstrate very impressive behavior. So today we have gotten about 1,000 cycles of SOL with 100% depth of discharge and a 90% retention. In a laboratory scale cell, it's quite small right now, but still exciting to work on. And we're looking to scale that. So the working principle of our technology is quite conceptually simple. The main ingredient is actually table salt or sea salt, a very common material that we all probably have in our own homes. From high school classes, we learned this to be sodium chloride. Now, on its own, it's quite stable. Most of us touched it with our own hand before, but it's also not conductive at all. In other words, it is entirely useless as a salt electrolyte for a battery. So we have to make chemical changes to it. So what we do is we first take this material and we dope it with some kind of aliovalent substituent or a transition metal with a high valence state. I'm just using an example such as zirconium 4+ or yttrium 3+. Now, because they have very different charge numbers, this will cause a charge imbalance in our entire system, and we are forced to remove some sodium in order to balance this charge. In other words, just by doping this into our sodium chloride structure, we have created vacancies in the structure, which now the sodium can diffuse. Yes, so the entire material now becomes a highly conductive material, and because it is very disordered, it's quite glassy in nature. That means the whole material becomes an amorphous glass. Now we've optimized this process over many iterations, and we can now fabricate it in a quite simple single step using room temperature processes. So we don't have to anneal anything, we don't have to heat anything to very high temperature, and we can use low-cost materials starting with table salt to produce this. So this invention has been patented, and we obtained some license from our home institution in California. And we'll also publish this in some reputable journals in order to be as transparent as possible to demonstrate this proof of concept. Now, although we are using a new material, we are not changing the way batteries are made. Our salt-based solid electrolyte, along with other components of the sodium battery, including your anode and your cathode, can be made using existing scalable manufacturing processes. And this is often regarded as the roll-to-roll slurry coating manufacturing process. And very similar to how lithium-ion batteries are being made, with the small modification that we incorporate the solid electrolyte inside instead of a liquid electrolyte. Now we can fabricate these full cells using tools that are currently available in the market in order to shorten the time towards commercialization and adopt the manufacturing process that you see in this diagram. And at the end, each layer is then stacked on top of each other to form a dense pack, or what we call a prismatic or pouch cell, in order to go into the battery product. So solid-state batteries cannot be, because they're solid in nature, they are ideally not rolled up into what we call a jelly roll. So you will not see cylindrical solid-state battery cell. Most of them will be prismatic or pouch format, similar to what you have in your mobile phones. So how does this technology compare with others out there in the market? So this plot here on the left compares gravimetric energy density versus volumetric energy density. Now, gravimetric, or in other words, watt-hours per kilogram, considers how heavy a battery is, or rather how much weight it takes per unit of energy. And in volumetric, instead of weight, it considers how much space it takes up. Well, because lithium is lighter than sodium, clearly it is going to be higher in gravimetric energy versus sodium. But there are many different applications for different type of battery chemistries. When you deal with power tools or starters, you would use lead acid, sometimes nickel metal hydride based chemistries. And when the device moves, like drones or planes which fly, or electric vehicles that have to move mass around, you want them to be as light as possible instead, which is why most of them use lithium chemistries. But for utility scale storage, it is stationary, it doesn't move, and we're going to put them in homes and businesses and things like that. So we are less concerned with how heavy it is, but how much space it takes up. To use an example, when we buy household appliances like a refrigerator or television, and when we go shopping for it, we don't really ask how many kilograms the refrigerator is or the television is. We instead, or how many inch is this? How much vegetables can I fit in this refrigerator? In other words, we're concerned about space because these don't move. So looking at volumetric energy density alone, we believe that the sodium solid state battery is very suitable for utility scale storage. And yes, and we will discuss this in further slides. So the stackable format that I talked about earlier is envisioned to be our first generation standalone product. We are still in the development phase and have not produced a product yet. The sodium solid state battery system will also be modular in nature and can be scaled to fit different kind of end user needs. So we expect this to be coupled with solar power generation, to be backup power in businesses, to go into 5G towers, and also serve as emergency power supply in disaster-prone regions or regions that are far away from the existing grid. And when widely adopted, we expect to offer end users utility savings, the ability to use renewables locally. They can also avoid peak pricing and reduce occurrence of power outage due to extreme weather conditions. So our first customers are expected to be existing energy storage providers, people like Shell and LG Chem, who we are currently jointly developing the technology and have been doing so for the past 3 years, along with other renewable generation companies who are keen to combine their generation systems with our storage technology. Beyond that, we are moving up in scale, moving away from homeowners to local microgrids and also to the larger grid. Homeowners always want to have utility savings because they want to avoid paying prices when it's most expensive. The grid, we also want to regulate the peak, especially if we are generating a lot of renewables locally. And finally, we also want to look at off-grid applications because when electric vehicles start to dominate the market, they also need charging facilities. And most of the time when you are out of the city, you need standalone charging facilities in the middle of the interstate freeway, for example, and those need to be powered by batteries. Otherwise, you have to spend a lot of money to build electrical infrastructure in the middle of nowhere. So the value and end users and utility providers will gain from distributed and stationary storage is threefold. Firstly, is peak shavings. Now, we always want to rely on more renewables, but we cannot simply install them and call it a day because renewables are on its own is intermittent in nature, right? The sun doesn't always shine. It's never always a sunny day. Unlike gas power plants or coal power plants, we can get a consistent energy baseline. The demand and supply curve of renewables is always fluctuating. But in particular, the demand curve, sometimes we call it a duck curve because it looks like a duck. This here shows the energy consumption in California on a typical spring day. As more and more renewables are installed, this valley starts to deepen more and more because during the middle of the day, we are generating too much renewable energy with nowhere to go. So currently about 1/3 of all renewable energy generated in California is just wasted. We send it to a load because there's nowhere to store it. And conversely, in the evening, when everybody come back home and start to use the television or start to cook and start to turn on heating and things like that, demand start to spike and generators need to work extremely hard to meet this demand. So this is a problem because this means that there's a lot of service interruptions and unhappy customers because of peak pricing. You can see the prices throughout the day fits exactly in this pattern. So there are of course short-term solutions touted by the utility companies, things like changing your time of use rates. They are encouraging people to take shower at 3 p.m. in the day instead of after dinner. They encourage people to watch TV in the middle of the day instead of at night, but not many people can do that, right? So by installing stationary storage in homes and locally, they can reduce the strain on the grid. Secondly, the other benefit is that of transmission losses. So this varies from country to country, but in the United States, about 5 to 10% of energy generated is lost on transmission alone. Now, that may sound like a small number, but it's actually incredibly significant because if you're going to generate giga-terawatts of energy every year and you're going to lose 5 to 10% simply to nothing, it's going to be such a waste. That's because humans do not always live in the sunniest of places like deserts or the windiest of places like mountainous areas where solar and wind power are generated. So we do need to transmit this energy over long distances and incur the losses. Additionally, building all this infrastructure, it can cost billions of dollars as well. So if we can distribute the storage and generation to where humans live, we can rely a lot more on the local microgrids instead and offset some of the initial capital costs. Thirdly, is a topic that is very more relevant, I would say, in recent years, is that of grid or energy resilience. Most of the electricity we use today is built on infrastructure that's almost a century old. It is extremely fragile in nature, with the Earth subject to stronger effects of climate change. We see increased occurrence of extreme weather conditions, things like wildfires and hurricanes. And this is especially important for offshore or island nations such as the Bahamas or islands in Southeast Asia as well. So we know the problem and we have the solution, but why aren't we in the market yet? Right now, our first generation chemistry, although it has been validated, there's still a lot of challenges with scaling and prototyping. In general, we face two types of major challenges in the near term, one that being of material supply and manufacturing. Unlike lithium battery materials, sodium battery materials are new and therefore not commercially available. We can't buy them off the market because no one makes them, despite the large abundance of sodium raw materials in most parts of the world. Therefore, we have to make all these materials ourselves from scratch, which we currently do on a very small scale. But we're still looking for resources to increase it to the kilogram scale and move away from the single layer prototypes that you saw in the video earlier, the multi-layer high energy cells, and all these things take time. We also need to expand our manufacturing capability. Currently, everything is somewhat still made manually. We have to move towards automation to increase output and this kind of thing. And we're also trying to expand the team and increase tooling capability on site. Secondly, we also have several issues regarding how we can deploy or penetrate the various grid markets around the world. Because we are not simply putting a consumer product in people's hands, they need to be integrated into existing infrastructure. And that forces us to deal with different kind of private or public agencies. We have to overcome administrative or regulatory hurdles unique to each state or each country. To elaborate a bit more about scale, these challenges are a testament to the harsh realities in scaling any form of deep tech. I would say the general public or technology investors in general have become quite accustomed to the exponential increase in software or digital information and computer processing power, which is famously characterized by Moore's law. On the other hand, unlike consumer commodities or this kind of software technology, the relatively sophisticated hardware and intensive capital tooling involved in things like battery development will ultimately result in slower pace of product development. In the end, it boils down to the fact that computers or softwares move electrons, while energy materials like batteries move ions instead, which are much larger, much slower, and a lot more sophisticated to handle. As a result, we see a very large gap in lab scale R&D, seen on the left here, versus industrial manufacturing at the end. Laboratory scale research typically led by academic and scientists focused on material level selection, testing and development, and the large scale focus on defect detection and also high throughput. Very few labs in the world are focusing on the center, which do pilot scale testing. And this is the main area where startups are focused on. Before I end, my last topic is that of battery sustainability. This is a up and coming topic for many battery manufacturers and battery scientists around the world, because the battery waste is starting to grow dramatically. To put things in perspective, the United States today has about 270 to 300 million combustion engine cars, of which 99% of its lead-acid batteries are recycled. So the lead-acid battery industry has done an excellent job over the past few decades to ensure that no matter how toxic, no matter how scarce, how expensive lead is, it is still very sustainable. They can achieve closed loop usability of their technology. In contrast to the many electric vehicles on the road today that are growing very quickly, recycling or collection rates vary drastically across the world. Some are as low as 5%, some are 50% collection, but still much lower than their lead acid battery counterparts. And with more and more electric vehicles coming back over the next 5 to 10 years, these batteries will reach the end of life and need to go somewhere, or else they'll simply be accumulated as waste. And in the 1970s, the US and many other governments around the world jumped into nuclear technology without thinking about what to do with the nuclear waste at the end of its life. So today we are dealing with the waste storage problem. So we should not make the same mistake for batteries. As such, the US Department of Energy has mapped out 3 major principles of battery recycling. First is direct recycling, which seeks to recycle batteries without breaking them down into their raw materials. And they also want to recover other materials in the battery beyond just the cathode, so that we can increase recovery fraction as a function of the entire cell. And thirdly is design for recycling, which seeks to design batteries with recycling in mind instead of disposal. It's really ironic that we always have recycling standards and recycling symbols for things like plastics, metals, papers, but we don't have this for batteries, because the batteries itself are not designed to be recycled. When the manufacturers make it, they do not think of it or what happens when it reaches its end of life. But doing all this is really challenging because it's not easy to convince them to change their current ways. But it's important for us to avoid the same mistake for next generation batteries, such as all solid state batteries, and ensure that they are sustainable before they even hit the market. So conceptually, the approach we take to recycle solid-state batteries are much simpler and easier than lithium-ion batteries. Firstly, because it's intrinsically safe, we can prevent sudden fires from occurring, so workers who handle them can face much less safety risk. It also reduces the cost of transport because it's no longer classified as a hazardous good, and the cost of transport can be decreased by orders of magnitude. And because there are no salts, separators, there are no liquids, there are much fewer components and we can potentially recycle close to 100% of all the materials, all without using toxic chemicals or energetic processes. We then design a protocol based on direct recycling in order to separate, recover, and regenerate both the solid electrolyte components as well as the electrode, and put them back into a fresh cell that can achieve, or rather already shown to achieve, the same performance as its original state. Now, because there are much fewer steps compared to conventional recycling, the modeling tools tells us that this design consumes much less energy and also emits much less greenhouse gases per kilogram of batteries recycled. To briefly summarize our team members, actually our company was founded at the start of 2020, and although we've been developing the technology for quite a number of years, we decided to commercialize it only quite relatively recently. We have 4 management members right now, and with Dr. Zeng Chen, Dr. Shirley Meng as academic co-founders at UC San Diego, and myself and Dr. Eric Wu running day-to-day operations and leading the development work. So with that, thank you for your attention. And if you'd like to learn more about the chemistry and how solid-state battery works, I encourage you to watch this YouTube video, which we put out there. And if you're interested to learn more about the battery development and the kind of research activities conducted at UC San Diego, I will recommend this video that you have here. With that, I will conclude my talk, and I'm happy to take any questions that you might have.
Thank you, Dr. Tan, for such deep insights regarding solid-state batteries. I would request the students to come up with the questions. I have a question in the chat box. Dr. Tan, am I audible?
Yes, I can hear you.
Yeah. There's a question. Is lithium ion or sodium ion battery 100% biocompatible?
Biocompatible? That's a question I haven't gotten before. I think the question tries to ask about the ecological footprints, possible ecological footprints of a sodium ion battery, probably. Ecological, all right.
Or I mean, what are the worst environmental impacts that this can have?
Oh, okay.
Theoretically or hypothetically, whatever.
And that's a good question. So lithium ion or sodium ion batteries, which conventionally use liquid electrolytes, I think the biggest problem is the toxicity of their materials. Because in lithium ion batteries, we use things like organic solvents, which are already toxic in nature and also flammable. And most importantly, the salts are fluorine based, so many of the fluorinated salts, when they get into the environment, is really damaging to both human health as well as the environment. And in sodium ion batteries, although we avoid many of the more dangerous transition metals in the electrode materials, the salt itself is still very dangerous. So I think if we want to be more environmentally friendly and biocompatible, we have to move away from this kind of toxic chemicals. Unfortunately, these toxic chemicals give excellent electrochemical performance, which is why many companies still try to use them. But if things move towards solid-state chemistries, we can completely eliminate the use of such materials. In fact, all our chemistries are entirely fluorine-free, and at the end of its life, they can be processed and safely handled using safe solvents. In fact, one of the main solvents that we use to handle them is ethanol. We've all familiar with ethanol. It's a main ingredient in one of my favorite beverages, which is beer or alcohol. But I would say that moving towards solid state can allow lithium and sodium ion batteries to be both safer and more, as you say, biocompatible.
There's another question. A very renowned, just a minute, let me move someone. Anyway, there's another question that a very renowned smartphone maker, I'm not naming the company here, although it's written in the chat box, recently flaunted about their air charge technology. We have already seen wireless charging for smartphones, but isn't it an astronomical development in the field of battery charging that your device can be charged completely wirelessly by just walking inside the room? What's your point of view regarding this and how it's even achieved?
So wireless charging is or rather, it's a continuously explored topic both by battery OEMs and also device makers. It brings about astronomical convenience to users and things like that. When people drive electric cars, it increases the value for consumers so much more when they don't have to think about plugging. Every time they park, it automatically charges. As a really, really good idea. However, the main challenge with wireless charging now is how energy efficient it is, and also the complexities in designing the inductors for wireless charging. So if you want to charge something wirelessly relatively quickly and also relatively efficiently, you have to put your device quite close to the charger. So for example, when wireless charging phone now has to be placed on the table right above the charger, because that's where the induction coil is. If you move it further away, I would say the charge rate decreases exponentially in a logarithmic scale. So right now there is still no I would say practical way to charge them while you are moving about, unless you have a very large I would say induction current always running throughout the entire room. I'm not sure what the effects of that on human health and things like that, but it's definitely not energy efficient. So induction wireless charging has been explored in electric cars better because the car is a lot closer to the road. So if the charger can be placed under the car where people park, it's much better. However, it is still highly inefficient because the distance between them and the geometries that they have to deal with, the induction coils and things like that are still not economically viable, which is why most of the battery companies have opted for fast charging cables instead. So if you see Tesla charging stations, the cables are getting thicker and thicker and thicker because they're going from fast charger to supercharger to now a super ultra charger. I haven't heard the latest, but that seems to be the way to go instead. And we are still, I would say, a decade or more away from widespread wireless charging.
Hi, Darren. This is Avik. I have some few technical questions.
Sure.
Yeah. So thank you for the nice talk. I think it's very important for the battery community, especially for the solid state battery community. So yeah, I just want to know, you mentioned about the halide solid electrolyte, but if you consider the alloy based anode, even consider the sodium alloy, which is like antimony, which is a bit higher, like I think 0.5 compared to sodium. How does it
Yes.
how does it stable with the halide electrolyte, or you plan for the bilayer, like sulfide halide, like bilayer kind of cells? So what's your like opinion on this? Like, so does the halide solve the issue for both? I'm the cathode side, I'm okay with the halide, but what about the anode side? I mean, does there any halide which also satisfy the anode side, even a bit higher potential, the anode?
Thank you, Abik. That's a very relevant question. So I draw a simple schematic here just to illustrate. Maybe not everyone in the audience catch Abik's question. So Abik talked about the halide-based solid electrolyte that we are used. And the halide solid electrolyte has a relatively low, not low, but narrow stability window where it is stable at the cathode, but not stable at the anode. So although we can use the halide here, definitely, we cannot use it in the electrolyte where it touches this point. So to answer your question, we can use the halide all the way to this point where I'm shading over here. And we do have to put a stable, very thin coating layer right before it touches the anode. And that layer over here is a different solid electrolyte. And currently what we are using is a hydride-based solid electrolyte, which is extremely stable at the anode. So this is another one of the main benefits of solid-state batteries, actually, because in a liquid-based cell where the liquid flows everywhere in the entire battery, you have to use the same electrolyte for the entire cell. So the electrolyte have to serve both the anode and the cathode. But in a solid-state battery where the ceramic doesn't move, you can choose different electrolytes to serve different purposes.
Okay, thank you. I have one more question. Like, okay, so the capacity retention you have shown, right? I mean, is it a room temperature, or you cycle the cell at a particular high temperature? And the second thing is that what is the expected aerial capacity you are looking for when you are considering for the grease storage? I mean, is it kind of like the one we are considering from the electric vehicle? I hope it is not. Like, you can compromise the aerial capacity a bit lesser because you do not have any constraint of the space. So what's your opinion on that part?
So two questions. One is the issue of the capacity retention. So now we are testing everything 30 degrees. We are strongly discouraged from using elevated temperatures. I know most literature still report 60 or higher in solid-state batteries, but we are trying to keep it constant at 30 degrees and occasionally testing lower. But right now, I think 30 degrees is a reasonable standard. Regarding your second question, it's on the
aerial capacity, basically.
Yeah, so when we talk about aerial capacity, are you referring to the aerial loading, or are you referring to the?
No. The whole capacity you actually drive to your cathode, from cathode to anode. Like, is it like normally, like, we are expecting like 4 to 5, and right now we are expecting 6 for the very high energy density battery, right, with the lithium metal. So suppose, I mean, are you expecting the same values for the grease storage, or grease storage basically because you do not have any constraint? So that's an advantage that you can even lower down your aerial capacity.
Yeah. So the aerial capacity for solid-state batteries are not as sensitive. The performance in solid-state batteries are not as sensitive to aerial loading compared to liquid. Many of the high-loading electrodes today are made using a dry process, which we are currently adopting. So the region of loading aerial capacity we are dealing with is about 6 to 10 milliamp hours per centimeter square, and that is simply because the sodium materials are a lot. The density is a lot higher than lithium. So both the halide is a lot denser and also the cathode is a lot denser. So although the aerial loading is higher, actually the thickness is not as large compared to the lithium. So we can do about 6 milliamp hours or higher. In fact, when we make the electrodes using a dry process, 6 is very comfortable.
Okay, I have the last question. Sorry for many questions, basically. It's always good to learn from you. So the last question is, like you mentioned about when you have the solid-state battery. So the advantage of this battery system is that even the conventional lithium-ion battery, you need a complex battery management system, right? Where the voltage modulation, current modulation circuit with the proper cooling system. So it has to be run constant temperature, because it's a kinetically stabilized process. As you slow the kinetics, this lasts longer. But when you replace with the solid state, you say like, okay, you don't need any complex system. Even it goes the high temperature helps to run the solid state battery, even the higher efficiency. But the thing is, I mean, considering none of the electrolyte really satisfy the energy diagram, thermodynamic energy diagram of the decomposition. So the electrolyte decompose, right? So even it heats up, right? It's an uncontrollable heat. And then if it is heats up like more than 100, 120 or 130 or 140, maybe you're getting the high capacity, right? Because of the fast kinetics, but your degradation of the battery in the solid state should be also higher, right? So is it like a more complicated battery management system do you need? Because I mean, it helps to the high value, but conceptually it should degrade your battery also very fast. So is it really can totally get rid of the management system, or like, what's your perspective on that part?
So when actually the battery management system is a very interesting topic. In lithium-ion batteries, there's a lot of effort and a lot of sophistication used to ensure that the batteries are always between 0 and 45 degrees. But in solid-state battery, if we can widen that to negative 20 to maybe 80 degrees, then that decreases the level of sophistication a lot more. So of course, at 120, 160, even 200 degrees, and that's extremely dangerous, and we want to avoid that completely. But I think most humans will not be living at that temperature anyway. We do have to have some amount of thermal management to ensure that it never crosses 100 degrees, for example. But we can be a lot more flexible in the day-to-day operation, for example. But the concern with battery management system in solid-state battery, it becomes less sophisticated on the thermal management, but it becomes a lot more sophisticated on the pressure management. So because solid-state materials are made of solid particles coming in contact, we always have to ensure that they have good conformal contact with each other. It's not like liquid that will flow and wet all surfaces. So when we operate the cell, it has to be under a certain amount of either uniaxial or isostatic stack pressure, and that pressure has to remain constant within its operation range. And here we are talking about megapascals. You know, this is about 100 times higher than the pressure we apply in lithium-ion batteries. So I think the sophistication in the battery management system will go towards the pressure monitoring side rather than the temperature monitoring side. So things like mechanics, how we can apply variable force as the cell is being operated, because the cell is going to expand and contract as we cycle it, and over long term, what are the effects of it is still not very clear. So many of the OEMs are keeping this in mind, but they are waiting for the chemistries to be ready before we can test it in the full device.
Okay, and sorry, last question is that, so right now there is a huge progress for the aqueous-based lithium and the sodium-ion battery. So how do you feel like the all-solid-state battery is kind of a like in terms of the competition, where the safety and the management issue is like a much more easier, even in terms of the cost also. So what's your perspective on that part? Like you know, and also like the company, there's one company for the Michigan-based company funded by the Bill Gates, which is actually working for the aqueous sodium-ion battery for the, especially for the grid storage, which is I think 2 or 2.5 or 3 times higher energy density of the lead acid battery. And then how do you think that the research progress going on the aqueous-based battery with the high concentration salt, even with the lean electrolyte, how the people are developing the new chemistries, the all solid-state battery can compete for the grid storage?
Okay, that's a very good and relevant question. Let me pull up my pointer. So grid storage actually is, we often think of all types of grid storage and we lump it together. In fact, I use my background and we often think of grid storage as megawatt hour scale. But actually, I would say vast majority of grid storage applications today are actually in the kilowatt hour scale. In fact, most installations are still in homes and businesses. We have yet to have widespread adoption in the megawatt hour scale yet. So we do have to divide the markets into 2 regions. One is the kilowatt hour scale, which is the probably homes and businesses, and the other side is the mega to gigawatt hour scale. So I do agree that for aqueous batteries and for even flow batteries and maybe even liquid metal batteries, that some companies have proposed, where energy density is absolutely not a concern at all. It will be dominant. And where we are not trying to compete with this large scale. We are looking at very much distributed energy storage, where volumetric energy density becomes somewhat of a concern. When people have to put this in apartments, people have to put this in houses, and people have to potentially put this in offices, hospitals, and things like that. So I think what we offer above all the other chemistries is two things. One is, of course, energy density. We do have to have some competitive volumetric energy density compared to lithium ion. And I think 600 watt-hour per kg is sufficient for that. Secondly, what we want to offer is also calendar life. Because this type of batteries are operated much more frequently than the large scale, because when we talk about distributed storage, we're using every day. People are charging and discharging constantly throughout the day, whereas large scale storage is used, I would say, more intermittently whenever the entire city needs a backup power or whenever there's an emergency scenario. So this kind of batteries need to retain their charge more. In other words, the self-discharge rates need to be as low as possible. And that is where the solid-state batteries are a lot more competitive compared to any kind of liquid form, especially things like flow batteries. I don't have the self-discharge rate for aqueous batteries off my head, but I do know that solid-state batteries are one of the lowest self-discharge rates out there in the market. So I think when we think of grid batteries, there are many different chemistries out there that will play different roles at different scale. What we are competing with is not against the very large scale, but we are looking and focusing on the smaller distributed storage scale.
Yeah, thank you very much, and best wishes with your progress of your imaging. Yeah.
Thank you.
Thank you, Dr. Tan, for addressing the questions. One last question, that is, does using lithium-ion or sodium-ion lead to a resilient grid? And what about the battery waste regarding sodium-ion batteries?
So this is two-part question. A resilient grid is, I would say, any kind of storage locally will contribute to a more resilient grid. What we see happening in the United States over the last winter and also over the last summer was that millions of people lost power to their homes, and they were left out in the cold, or they were trapped by wildfires, and many of the utility companies had to shut down for safety reasons. So this tells us that our grid is actually not resilient at all. So I would say it's not chemistry based. Having more storage would improve the resilience of the grid. The question now is, what kind of storage technology are we going to use? The second question is about battery waste produced until the last decade. How can we dispose of it? Unfortunately, most batteries today are not recycled because it's too expensive to recycle them. It's actually cheaper for the companies to rent space and store the batteries. So many of the electric vehicles that come back, all these packs are being stored in warehouses, and they're patiently waiting for the recycling technology to catch up until one day it is cheaper than to recycle it than to store it. There are also other applications people are looking at, Second Life, where we can repurpose it for other purposes, but that also is a fairly complicated undertaking. At the end of the day, I think we must improve our entire battery recycling infrastructure, from the way we collect it, from the way we transport it, who is going to handle it. In my opinion, some degree of responsibility has to be shifted towards the original manufacturer in the first place, because currently most batteries are manufactured in Korea, China, Japan, and they are being used in other countries. So the problem shifts away from these original manufacturers towards the United States, for example. I think that it is a trend where, because raw materials are becoming extremely scarce, there is a recent topic of critical metals meaning like lithium, cobalt, nickel, and this kind of thing. And with more and more batteries moving away from where they're being mined to where they reach the end of life in other countries, the shift in availability of critical materials will start to change. In other words, in some point in the future, we will view used batteries not as waste, but as raw resource to mine critical materials from. And that will be one of the main economic drivers for battery recycling. And I really hope that happens soon.
Yeah. Thank you, Dr. Tan. So thank you again for cooperating with us. I know the time at US is not really, I mean, it's more or less an unearthly time, but still you cooperated, you and Dr. Bhattacharya, both of you, just across the globe.
I'm in the West Coast, so it's perfect breakfast time for me now.
That's great. So now I would like to call Professor Sonjeev Ghosh for the valedictory session. Sir.
I just heard the lecture and it was a nice, beautiful lecture. Can you hear me? Prantik?
Yes, sir.
Yeah, this is a nice lecture and
Yes.
very wonderful, and lots of information and things like that. And I don't know whether the sodium-ion batteries, whether it will be, some batteries will be biocompatible or not. Whether these sodium-ion batteries could be biocompatible.
I'm sorry, did you say biocompatible?
Yes.
Oh yeah, actually one of the audience member asked a question. We also answered it. So that depends on the very much on the electrolyte chemistry, because we elaborated that liquid-based sodium-ion and lithium-ion batteries often use toxic and somewhat dangerous lithium salts and solvents, and also sodium salt and solvents, and these are things we want to avoid. Those are definitely not biocompatible. But moving forward, if we adopt a salt-based chemistry and ionic ceramics, it would definitely be a lot more environmentally friendly and biocompatible, and we can also recycle it as well.
Thank you.
Sir, this is now we are going to start the valedictory session. I request all my speakers and those participants to stay for a while, little bit. So, sir, please, Professor Ghosh, sir, please start the valedictory session. We want to start with you on.
What about the other lecture? That will not be possible?
No, actually, sir.
The second lecture was somebody.
There is a problem, actually, sir. He could not join, actually. So this is the end of this.
Okay. So the final session, I just learned a lot, I should say. And first of all, I thank Dr. Arpan Mandal for organizing such a wonderful seminar, which is one of a kind, because it's a link between academia and industry. I think students, faculty members, and researchers got a lot of information regarding how to get to industry, how to from academy, or after their degree in pure science, they can move to various fields like biotechnology, biochemistry, molecular biology, microbiology, starting from that to sodium-ion battery or lithium-ion battery and energy sector and many various sectors they can go. We heard a very nice lecture about fiber optics. So lots of possibility. There is a lecture AR and VR. We didn't know about those kind of things. So it's a very wonderful session, all these sessions, and I think I have learned a lot. So I think students also get benefited, and faculty members too. And in this pandemic period, at least 2 days, we could forget about pandemic and listen to some topics which are so interesting. And I just pray that this COVID situation will be over within few days or few weeks. And during this period, I just advise all faculty members, students, everybody to engage themselves to something, to read a lot or to do something creative if possible by staying at home, and try to get expertise in some field apart from your own field. Like chemistry, you can read physics, or you can read some mathematics, you can read some biology or biochemistry, something like that. So this is a period when you can learn a lot of things, and all the speakers are so nice that they already told us that whenever you want, you can connect them. So they have Orpon has all the email ID of all these speakers. So if someone is interested in some particular field, he or she can connect Orpon and connect directly to the speakers and get more information, more ideas about how to get to the industry from academia, from after passing a BSc or MSc degree.
Thank you very much, sir, for your wonderful inspiration. And I wish to invite at least two of you, the participants, to share their experience regarding this 2 days journey of this international symposium. Anybody can unmute and share your experience.
Thank you, Orpon.
Thank you, sir. Any of the students or anybody, any participant, please share your experience. Just unmute and share your experience regarding this 2 days talk. Mr. Roji Banerjee.
Our MSc students are there.
Yes, they are those there. Roji Banerjee, can you share your experience? Actually, she had a lot of questions for 2 days. She is completely involved. Rosie, please.
Hello.
Rosie, please.
Can you hear me, Dr. Arpan Manna?
Yes.
Yeah. Thank you for inviting me for this symposium. And yes, I have had a lot of questions because I have been out of touch with my subject for a few years because I have been deviating from my field, and amid this pandemic, I have been going through certain things, but it felt like I was detaching from it. But these 2 days symposium has made me feel like I'm really alive. I'm really, really alive. And listening to all the speakers and all the lectures in these 2 days, felt that the world is progressing a lot and we need to do a lot more from our side also. Because there are things, yes, obviously there were things which was like, bouncing my head because I had no idea about certain topics. But yes, I tried to get connected because Dr. Arpan Manna was my senior when I was pursuing my MSc. So I call him Mannada. So he had sent me a PDF which comprised of all the lectures which were being delivered for these 2 days, along with the topics for the speakers. And I tried to search and I tried to gain some knowledge about it. And while doing these, while going through this process of searching about things and gaining knowledge about things, I gained a lot of experience about it. And I wish to do the same for if it's possible in case. So it has really been very helpful for me, like regarding this COVID pandemic, when Dr. Bhattacharya was giving her lecture this evening. So I have had a lot of questions because I have been through YouTube videos. I have been through many newspaper articles like this is being done, this is being done. But what the original scenario was, it's very difficult to get it to know through the media outlets. So it was very helpful from her side to know that, to let us know that what really happens and where we are standing in this particular world. So it has really been helpful from everyone. Thank you so much. Thank you so much, Mannada.
Thank you, Rosie. So anybody else? Anybody else? Anybody else? Dr. Puspa B, can you share your experience? Dr. Puspa B, are you there?
Ask our student like Madhuparna is there, I think.
Madhuparna, that your experience? Madhuparna,
Yes, sir. Yes, sir.
tell me your experience.
Yes, sir. It was a great experience. I've learned a lot from all the speakers, and it was my pleasure to hear them. And I'm very lucky. I feel very lucky to hear such great person.
Thank
So
you.
it was, yes, sir. Thank you for organizing such a great symposium. I'm very pleased and very satisfied.
Okay. Uh, so we are almost coming to the end actually. So uh, I am thankful to all my faculty members from the Department of Chemistry. I am thankful to Professor Sanjeev Ghosh. He is the source of a constant inspiration to all of us, and he is the pioneer to keep our Department of Chemistry as vibrant. Because he's actually, you can think of that he is the grandfather in the field of physical chemistry. Lots of great scientists, academicians, as well as some professionals have been taught by him. And also I am thankful to our Honorable Chancellor, Sir, Professor Sumit Ray. And I am also thankful to our Vice Chancellor, Sir, Professor Jitendra Kumar Jha, Sir, for their constant inspiration and also giving us this wonderful platform and giving the permission and all these things. I am also thankful to our Dean, Sir, Professor Jitendra Kumar Pandey, Sir, which also who had actually he who had a background of working on the polymer field, and he had done his PhD from National Chemical Laboratory. And Pandey sir, after joining in our university, he is inspiring us on every day. I can think that every moment that Arpan, we have to do some new things that our student should not suffer, because this is really a very hard situation, hard condition, pandemic situation. Most of the students, especially the first year students, we have not seen them face to face, and we have not taken not a single physical class for last 1 year, some months actually. So still they are bearing with us. They are doing the online classes regularly, and I know that there is a lots of problems for doing online classes, because I as by my experience, the doing this type of online classes without this physical contact or physical classes, it's very difficult to understand that my student really he or she had understood or not. And another worry that one of our participant, she had told that we have not gone to the laboratory for last 1 year, both for you undergraduate and postgraduate student. What can we do? Really, it is a serious thing. And we thought that we can do the more laboratory courses in this year, but the situation again, it is not okay. So we have to think some new thing, that our student should not go pass their undergraduate or postgraduate courses without doing a single lab class directly. So I would like to express the sincere appreciation and gratitude to all the coordinators, presenters, and the discussion and the participants for your very active and frank discussion. And I think all of you have made this conference, this 2-day symposium, very fruitful. I will start with a word by John Days, that education is not a preparation for life, but education is life itself. So as a teacher, we have to deal very sensitively with this constantly evolving aspiration of the student. Why I am stressing on this word aspiration? Because from the childhood, each of our family aspires for the good quality of education for the child, especially a country like India, because every child in this class is a bundle of aspiration, which ranges from a simplest one during early childhood to more evolved one in the later cases. We have also heard Subhajit Chatterjee in the afternoon that our aim constantly changes with time as we grow up, as we learn new things. So same thing, our aspirations also changes with time. But this aspiration is the starting point of the journey to the achievement, both for the individual as well as for the nation. So I am thankful to all the distinguished speakers. Now, Dr. Ovik Banerjee, Dr. Samosri Bhattacharya, and Dr. Darren Tain here with us. So your presence is really a great motivation to all. Throughout these 2 days, we have been so impressed by the eagerness among our presenters as well as the participants and their co-operators. I am sure that we all had 2 days of very productive discussion. Given the very distinguished list of speakers that I have seen and my participants have seen, that even more distinguished gathering of participants from the various corners of the country. So last but not the least, I want to thank all my participants as well as all my distinguished speakers and my faculty members, both from the Department of Chemistry and also the faculty from the Environmental Science for their constant touch. They also have toiled a lot to make this event successful. So thank you all. And another one announcement that the feedback form, I will mail you by tonight to all of you. Just fill this feedback form, and as soon as you fill the feedback form, so since there are a lot of participants, almost 250, so it will take 2-3 more days you will receive your participation certificate. Thank you very much again. So be safe and thank you all.
Thank you.
Good night, everybody.
Good night, sir.